Hi, my name is Jennifer Gray. I'm a TEM staff scientist here at Penn State. I work in the materials characterization lab on our transmission electron microscopes, and we are part of the Materials Research Institute. So, what I'm talking about today is how we can use a transmission electron microscope to image delicate materials that damage under the beam very easily, or hydrated materials which have some sort of water in them or other liquid that you wouldn't necessarily be able to put in the vacuum environment of the electron microscope otherwise. So first I want to give a little bit of background on the transmission electron microscope and the types of information we can get from it. So to look at the sample, we're going to use a high energy electron beam, usually between 200 or 300 kV, and that beam needs to pass through the sample and be scattered by the sample so that we can see an image on our camera that's going to give us contrast based on either mass or thickness differences in the sample or diffraction effects on the sample. So regions that are thicker or have heavier mass are going to appear darker. Regions that are strongly diffracting might appear darker. And so this can give us a lot of information about the sample all the way down to a resolution of 1 angstrom or even less in some of our more advanced microscopes. But not only do we get an image of the sample, it's also telling us about the crystal structure of the sample. If we look at the diffraction pattern, if we look at the X-rays coming off of the sample, it can tell us about what the composition of the sample is. We can get an elemental map. Again, we can get high resolution images showing the actual crystal structure of the sample and how the atoms are arranged. We can also look at the electrons that have lost energy to the sample, and that is called EELS, or electron energy loss spectroscopy, and that can tell us about the oxidation state of the sample or the chemical bonding, which will be information that's kind of analogous to what you would get in XPS. We get a lot of information from TEM if we can get the sample into the microscope and if the sample can survive under that high energy beam. So those are kind of our constraints, and what I'm talking about here is the different methods we can use to get around those constraints. So when we're talking about constraints in TEM, what I'm talking about are kind of the historical limitations that we've had when doing transmission electron microscopy, and that first of all, your sample has to be able to be stable in a high vacuum environment, meaning it can't be liquid, it can't be hydrated, it can't be sticky. And second is that the sample has to be able to withstand the very high energy or high dose we're generally giving it with the microscope. So if a sample damages immediately, you're not able to see the pristine structure of the sample. And again, if the sample is going to dry out before you can put it in the microscope, you may not see it in its native environment. How are we going to get around these limitations? The first thing we use is some newer technology, which is called a low-dose direct electron detector instead of a traditional CCD camera. These were developed initially, I think, for a lot of the bio-EM applications where they were looking at samples that would damage very easily, like viruses. And so we're going to adapt these now for material science applications when we have very beam sensitive materials. And so how they're going to work is they're going to give us a lot higher sensitivity because they're directly converting the electrons that come through the sample and hit the detector, as opposed to the old CCD camera technology. You would have those electrons would be converted into an optical signal and then back to an electronic signal. So these give us a lot better sensitivity so that we can work with much lower doses than we would have been able in the past and still get an image that doesn't look completely noisy. The second thing we can do to try to limit the damage in a sample is to use a cryo holder. So that means a holder that can keep our sample at liquid nitrogen temperatures so that we can slow down the damage that may be happening when it gets hit with the electron beam. So on the right-hand side of this slide, you see this a cryo holder right there, which is basically the back end of that is a little liquid nitrogen dewar. And so that keeps the sample under liquid nitrogen temperatures all during the imaging, and that can help slow down the damage process that these samples go through underneath the beam. And then finally, if we want to look at samples that are hydrated or liquid samples, we can use a special type of thin window holder, where the liquid is sandwiched between two very thin silicon nitride windows. And so these windows kind of encapsulate the sample and protect it from the vacuum, but yet are electron beam transparent. So the electron beam can go through the windows, can go through the sample, come out the bottom side, and you're able to image a liquid sample without ever having it actually see vacuum. So this slide shows an example of what I mean by low dose imaging using a direct electron detector, this newer technology that's a very sensitive type of detector. So on the left-hand side, we have an image of a very thin boron nitride flake, a crystalline boron nitride flake, where we've imaged it using only a few electrons per angstrom squared on the K2 direct electron detector. And then we can compare that using the exact same dose if we image this sample on a traditional CCD camera. And so on the traditional CCD camera, you just see basically noise. You don't really see that periodic crystal structure in the image. Where on the left-hand side, the DED direct electron detector image very clearly shows the regular arrangement of the columns of atoms in that crystal structure. And the upper right-hand corner just shows the FFTs for these two images, so you can see how much clearer the FFT is for the direct electron detector image on the left as opposed to the one on the right. This is another example of low-dose imaging now done on a 2D zeolite nanosheet. This was for catalysis applications. And again, this type of material, it would immediately damage underneath the beam. You wouldn't be able to necessarily get these nice high-resolution images showing the lattice fringes in the sample under our standard dose conditions, but under these super low-dose conditions using a direct electron detector, we can see the lattice fringes showing us the structure of the sample. We can count the number of sheets in the sample to kind of understand the thickness better. So again, this type of information won't be possible using our standard dose conditions on a traditional CCD. This is another example of a semiconductor polymer crystal that would be used for organic electronics applications. And again, using a direct electron detector, in addition to cryo conditions, we can directly see the lattice fringes in these images and show that these polymer nanoparticles are actually crystalline. Beyond just using these new direct electron detector cameras as well as cryo-imaging conditions, the other thing we've borrowed from life sciences in order to minimize dose on our samples and look at these really beam-sensitive samples is special low-dose data collection software. This software that we're using is Thermo Fisher software called EPU, which again is commonly used in the bio world for collecting thousands of images in a semi-automated kind of way. So what we can do with this software is we can take a really low mag image of our entire grid square, and then we can drive to specific squares within that grid square while the beam is blanked. We can go to the grid square of interest or maybe a sacrificial grid square. We can do our focusing. We can check our alignments. And then we can, again, blank the beam and go specifically to the real region of interest and collect an image that's now focused and at extremely low dose conditions so that it's never really been exposed to the electron beam in any significant manner before the image has been taken. And so as opposed to in the past without this kind of software, we'd have to drive around with the beam exposing the sample the whole time until we found that region of interest and try to take a picture as fast as we can. This is a way that we can use this software to facilitate giving us a much lower dose on the sample before we take the picture. So now I want to talk about liquid samples. What if we have a sample in liquid, like maybe nanoparticles suspended in liquid, and we don't want to dry it because maybe that's going to change the properties or cause agglomeration or something like that. So we want to see how it behaves in liquid in the microscope. So the way we're going to do this is we're going to use a Vitrobot to plunge freeze the sample in ethane. And when we plunge freeze the sample in ethane, we're going to basically put a droplet on a TEM grid and then flash freeze it in the ethane. And that means it's going to freeze so fast, it's going to form amorphous ice and not crystalline ice. And so that's going to give us a nice imaging condition to see that frozen sample. So that's one way to do it. Another way is to try to image the liquid directly as long as we can protect it from the vacuum of the microscope. And again, there's a couple ways that people do this. One is to use silicon nitride chips to basically sandwich the liquid between two chips and use a special holder that's designed to be able to use these chips to image with. So basically your windows are so thin that they're electron beam transparent. And so the beam is able to travel through the silicon nitride barrier, through your sample, and out the other side of the second silicon nitride window. And so again, you're able to then image the liquid without the liquid ever seeing the vacuum. This is a great method to use, although you're going to lose maybe some resolution due to the thickness of those silicon nitride windows. So another way you can try to do it is encapsulate the liquid between two graphene layers. This would be for static liquid samples usually, whereas with a silicon nitride liquid holder, you can actually flow liquid into that as opposed to just doing static liquid. But with the graphene sandwich samples, you have a very thin layer of graphene that's going to maybe let you go to get better resolution because you're not going to be affected by that thicker silicon nitride window. So this is one example of cryo-TEM imaging of frozen polymer latex particles in solution. So these were in a solution that was frozen using the Vitrobot, so very quick freezing, so that these particles that you see are basically encapsulated in this amorphous ice. And so again, we get very nice images of these particles using this method. And then the second method that I talked about is the in situ liquid holder. We use a Protochips Poseidon holder here at Penn State. There's other manufacturers as well. But again, this is two silicon nitride chips that sandwich the liquid in between them. You can see a diagram on the right-hand side showing that we have this O-ring, and then a small chip and a large chip, and the liquid is basically in between those two gold-colored chips. And so we tighten the lid down, and so that keeps it all sealed up, again, so that liquid can't escape into the vacuum of the microscope. And again, you can either put a droplet of liquid between those two chips, or there's little fluidic ports that you can actually flow liquid in, and so you can watch reactions happen in real time in the microscope. We also have the ability to heat the sample up to 100 degrees only in this type of holder if you want to look at some, again, reaction kinetics at maybe higher temperatures. We can also do electrochemistry. So there's a lot of potentially really interesting applications for in situ liquid imaging in the microscope using this type of holder. Although these experiments are not easy to do. They usually take a lot of time to optimize, a lot of trial and error. Again, it's difficult to image in thick liquids because you lose a lot of resolution. So there's some things you have to be aware of before diving into this type of method. One of these concerns would be, what does the beam do to the liquid? So this is work that was not done here. This was done by Taylor Wall's group, but it nicely shows the effect of how the beam can interfere with what you're trying to see. So the beam can basically create all these kind of free radicals in the liquid When it hits the water, it can dissociate the water molecules, causing all these charged species, which can then affect what you're trying to look at. So in this study, his group was looking at the nucleation kinetics of silver nanoparticles, and what they see is that as you change the dose, i.e., you go to higher magnifications, more dose, it's going to change the number of particles that are forming in a given amount of time under higher dose conditions. So you're accelerating the process of particle formation. So it's just something to be aware of if you are going to do liquid imaging and trying to actually study some process happening. You always have to be aware that the beam may be changing the environment that your sample is in while you're looking at it.